essential and non-essential roles of typical flower. This comes under sexual reproduction in flowering plants. So let us begin with the detailed account of male and female reproductive parts in angiosperms. The male reproductive part is androecium, and the members of androecium are called stamens or microsporophylls. You need to remember both the words. Each stamen has two parts, anther and filament. Filament supports anther. The point where anther is fused with the filament is known as connective. Now there are different shapes we found in the anther. It may be kidney shaped, it may be arrowhead like, it may be bilobed. There are different forms of anther. It is considered as fertile part of a stamen. So when we take transverse section of young anther, we can see anther wall and pollen sacs called as microsporangia. There are two types of anthers depending on the lobes. Monothecous with single lobe, dithecous having two lobes. When anther is monothecous, it has two microsporangia. So we call monothecous is bisporangiate anther, and dithecous having four microsporangia. So we call it is tetrasporangia. As per the syllabus, we are dealing with dithecous tetrasporangiate anther. The question will come for 5 marks. You should be cautious when you read the question. They may ask, explain the structure of young anther or explain the structure of mature anther. When it is young anther, ray diagram you need to give showing anther wall composed of 4 layers and 4 regions known as microsporangia. The 4 layers forming anther wall are outermost single layered parenchymatous giving protection known as epidermis. Second layer is 2 to 3 layered known as endothecium, which gives mechanical support. Middle layer is multi-layered, having parenchymatous cells. This acts like a shock absorber, also nourishes the anther wall. Innermost layer, which lines the microsporangia, is known as tapetum. So 4 layers forming anther wall. Outer one is epidermis, second layer is known as endothecium, third layer is known as middle layer, and innermost is tapetum. Now speciality of the tapetum is the cells are binucleated, having, or sometimes they have more than 2 nuclei also, and they contain dense cytoplasm. It is a nourishing layer which provides the required nutrients to developing microspores or pollen grains. So when you expose the anther wall at one side, we can see the typical arrangement of these cells. It is shown in the diagram. Now when we expose single microsporangium in young condition, it contains a mass of undifferentiated cells known as sporogenous tissue. When the process of microsporogenesis begins, the cells of sporogenous tissue start maturing. So the first stage is known as archisporial cell, which is diploid. This archisporial cell now differentiate as PMC or pollen mother cell, having 2n number of chromosomes. Now this pollen mother cell or microspore mother cell which undergo meiotic divisions. As you already know that meiosis includes two divisions and it is a haploid division. So meiosis 1 gives two haploid cells known as pollen dyad. Meiosis 2 gives four haploid cells which are together known as pollen tetrad. Now from this pollen tetrad further differentiation begins. Consider single cell out of these pollen tetrad. This single cell having an active functional vacuole and a big nucleus surrounded by the wall. Now the maturation begins. The nucleus undergo division. It gives big and small nucleus. The small nucleus floats in the cytoplasm of that bigger portion. Later, a compartment develops, a partition wall develops known as Callus wall between the big nucleus and the smaller nucleus. So at this stage, we can see the cell in two-celled condition. The bigger compartment is known as vegetative cell and the nucleus is said to be vegetative nucleus or tube nucleus, and the smaller compartment is known as generative cell and the nucleus is called a generative nucleus. So in microsporogenesis, so what exactly microsporogenesis? It is the development of microspore or pollen grain from diploid pollen mother cell. I'll repeat. Microsporogenesis is a process of forming haploid microspore or pollen grains from diploid pollen mother cells. The different stages, which are diagrammatically depicted as sporogenous tissue, gives archisporial cells. Archisporial cell differentiate as pollen mother cell. It gives pollen dyad after meiosis 1. It gives pollen tetrad after meiosis 2. And then all four cells separates and further differentiation begins. During this development, the bigger nucleus divides. It is endomitotic division, giving vegetative nucleus and generative nucleus. Between the two nuclei, a callus wall develops. Around this mature cell, now there are two-celled condition here. The outermost layer develops, which is deposited with a special chemical which provides resistance against heat, cold, known as enzyme, and that chemical name is sporopollenin. The inner layer, which is slightly flexible and elastic, is intine, made up of cellulose, sometimes cellulose with pectin. So a mature pollen grain represents microspore. It is a male gametophyte having two layers. Outer enzyme made up of sporopollenin, inner layer is intine made up of cellulose and pectin. It encloses two compartments as it is two-celled stage, vegetative cell or tube cell enclosing vegetative nucleus or tube nucleus, and the smaller compartment is generative cell enclosing generative nucleus. So at this condition, the pollen grains or microspores are ready to come out from the anther. Now, the next portion, when microsporogenesis is completed, the water content in the middle layer, endothecium, reduces. As the water content reduces, the pressure is exerted on the anther wall on both the sides. So, due to this exerted pressure, anther wall will break. This is called dehiscence of anther. So, when anther wall breaks, now it is a mature anther, after microsporogenesis to release the pollen grains or microspores for pollination. So when anther wall dehisces, a opening which appear like a mouth having two lips known as stomium. So when the stomium develops, forcefully all microspores or pollen grains are released outside. And now they are ready to undergo pollination. In this session, the important questions which are appearing in the board exam are: name the male reproductive whorl in flower. Answer is androecium. Members of androecium. Answer is stamens or microsporophylls. Diagrammatically represent parts of stamen. You need to draw a small diagram and label two parts. Filament is the style and fertile part is anther. Next, two questions appearing for 5 marks each. Explain the structure of young anther. Explain the structure of mature anther. If it is young anther, I have already shown in the diagram. Diagrammatic representation showing 4 layers forming anther wall. At one side you need to expose the anther wall to show the arrangement of the cells forming epidermis, endothecium, middle layer and tapetum. Here tapetum nourishes the developing microspores during microsporogenesis. There are 4 microsporangia in tetrasporangiate anther, enclosing sporogenous tissue. Clear? Now, the next question carrying 5 marks weightage is microsporogenesis. So here you need to represent serially on the diagrams like archisporial cell, pollen mother cell, pollen dyad, pollen tetrad, and then a mature pollen grain. This we need to explain, which carry 5 marks weightage. There is one more question expected. Explain the structure of pollen grain. A mature pollen grain you need to draw showing exine, intine, vegetative cell, generative cell, and when you explain this diagram, exine is outermost layer made up of sporopoienin which provide resistance against heat and cold. It keeps the pollen grains viable till they deposit on the stigma. Inner intine is flexible, it is made up of cellulose and pectin. Vegetative cell which will germinate as pollen tube to carry generative cell towards the ovary, which is future forming male gametes. So generative cell divides to give 2 male gametes. Here the most important point, exine is porous. There are minute germ pores. Through the germ pores, the vegetative cell develops into a hollow pollen tube when pollen grain deposits on the stigma during germination. Here, with this, we have already completed the male reproductive whorl androecium. Members are stamens, parts of stamen filament anther, 2 diagrams, young anther, mature anther, diagrammatic representation of microsporogenesis where you are showing archisporial cell, pollen mother cell, pollen dyad, pollen tetrad and a mature pollen grain. Last, the structure of pollen grain with all the labelings exine, intine, vegetative cell, vegetative nucleus, generative cell, generative nucleus and the explanation. With this we have completed the second session of the topic sexual reproduction in flowering plants. Thank you very much.